Hey, this is Rochelle. In today's microbiology interactive tutoring session, we're going to be discussing cell nutrients and macronutrients, micronutrients. How do the nutrients enter into the cell? We have simple transport and group translocation. In order for this interactive tutoring session to be interactive, go ahead, go to the description box and grab your workbook. So let's microbiology it. Cells require nutrients to grow and function, and prokaryotic cells obtain essential elements like carbon, nitrogen, phosphorus, and sulfur in specific forms. Carbon is sourced from organic compounds, which are large polymers that break down into smaller, usable monomers such as amino acids, sugars, fatty acids, and nitrogen bases. Nitrogen is taken up in several natural forms, including ammonia, nitrite, and nitrogen gas. Phosphorus is needed for building nucleic acids, phospholipids, and energy-rich phosphate groups. Sulfur is found in certain amino acids like cysteine and methionine, as well as in vitamins such as thiamine and biotin, and in forms like sulfate and hydrogen sulfide. In addition to these macronutrients, cells also require micronutrients like iron, which is essential in respiration, and other trace metals that act as cofactors. Organic growth factors such as vitamins, amino acids, purines, and pyrimidines are also required, especially by bacteria like Streptococcus and Lactobacillus, which often get them from nutrient-rich environments like the human gut. To bring these nutrients inside, cells use active transport mechanisms that require energy, including proton motive force, ATP, or other high energy compounds. There are three main active transport methods: simple transport, which uses a membrane protein and is powered by proton motive force, or PMF; group translation, where the substance is chemically modified during transport using energy from compounds like phosphoenolpyruvate; and ABC transport system, which uses ATP and consists of a substrate binding protein, a membrane spanning transporter, and an ATP hydrolyzing protein. In gram-negative bacteria, periplasmic binding proteins, PBPs, have high substrate affinity and help capture nutrients even when they're scarce. In gram-positive bacteria and archaea, which lack a periplasmic space, the binding proteins are attached directly to the cell membrane but function primarily or similarly to bring nutrients into the cell using ATP. Here is the interactive portion of our interactive tutoring session. Are you ready? I know you got this, so here are the instructions. Fill in each blank with the correct term from your study material. So we went over this. We know this. We got this. All you have to do is fill in the blank. Let's start with number 1. Prokaryotic cells use what? From organic compounds, which are broken down into blank substances like amino acids and fatty acids. Number 2. The three main forms of nitrogen found in nature are what? 3. Phosphorus is used in what? Number 4. Sulfur is found in amino acids like, as well as in vitamins such as what? Number 5. Blank is a trace metal involved in cellular respiration. Number 6. Trace metals function as what in cells, while growth factors are blank, compounds such as blank, blank, and blank. 7. Some bacteria, like Streptococcus and Lactobacillus, receive blank from nutrient-rich environments like the blank or blank. 8. Active transport is a blank-driven mechanism for importing nutrients into cells. 9. The three basic types of active transport are what? 10. In simple transport, the energy source is the blank. 11. In a blank reaction, a proton and solute are transported simultaneously in the same direction, while in a blank reaction, they move in opposite directions. 12. An example of a symporter is blank in E. coli, which transports blank along with a proton. 13. In group translocation, the transported substance is blank modified. The energy source for sugar transport comes from blank, a compound from glycolysis. 14. The blank system in E. coli phosphorylates sugar like glucose, mannose, and fructose during transport. 15. In gram-negative bacteria, blank proteins have high substrate affinity and assist in the ABC transport system. 16. The ABC system includes three components: the protein, a blank, and a protein. 17. In gram-positive bacteria and archaea, which lack a blank substrate binding protein, are attached to the blank of the cytoplasmic membrane. So that's the end of our first worksheet. Worksheet number 2. Here are the instructions. Choose the best answer for each question. So again, the answers are coming from the notes. So review your notes. You can pause the video, answer the question, follow along, whichever is best for you. So it's multiple choice. Let's start with number 1. Which of the following are monomeric products of organic compound breakdown in prokaryotes? Two, which element is not a common nitrogen source in nature? 3, phosphorus is primarily involved in the formation of. 4, sulfur can be found in all of the following except. 5, iron is an essential micronutrient because. 6, which of the following is a growth factor? 7, what drives nutrient uptake in active transport mechanisms? 8, which transport method modifies the substance during transport? 9, which example is associated with group translocation? 10, what component is not part of an ABC transport system? 11. In gram-negative cells, where are periplasmic binding proteins located? 12. How do substrate binding proteins function in gram-positive bacteria? And that's the end of our second interactive worksheet. Hey, this is Rochelle, your microbiology tutor. Welcome to another Microbiology IT interactive tutoring session. Today's video is molecular info and protein processing. So, what is DNA and how is it replicated? Who are the major players in replication? And how are proteins made? Well, in this video, we're going to answer those questions. And to make this session interactive, click the link in the description box to follow along with your ebook. Let's microbiology it. The molecular flow of genetic information and protein processing is central to understanding microbiology. At the heart of this process lies the genetic material, DNA, which serves as the cellular blueprint for all biological functions. DNA, composed of a sequence of nucleotides, is the genetic code stored in the double helix, a stable structure held together by hydrogen bonds between complementary base pairs. The strands of the double helix run anti-parallel, allowing for efficient and accurate duplication and transcription. Nucleotides, which make up nucleic acids, are composed of a nucleoside and a phosphate group. These repeating units give DNA its structure and functionality. Genes are specific sequences of DNA that carry the instructions for building proteins or functional RNA molecules. Genetic elements include both chromosomal genes and genes located on larger molecular structures such as plasmids. The complete set of an organism's genetic information is referred to as its genome. During cellular processes, RNA acts as the intermediary between DNA and protein synthesis. Three primary types of RNA are involved: messenger RNA, which carries genetic information from DNA to ribosomes; transfer RNA, which decodes the messenger RNA sequence into amino acids; and ribosomal RNA, which forms the catalytic and structural components of ribosomes. The flow of genetic information follows a defined pathway that includes replication, transcription, and translation. Replication is the duplication of DNA, a process catalyzed by DNA polymerases. Transcription involves the transfer of genetic information from DNA to RNA and is driven by RNA polymerase. Translation is the synthesis of polypeptides based on the sequence encoded in messenger RNA, ultimately resulting in protein formation. In eukaryotic cells, replication and transcription occur in the nucleus, and messenger RNA is exported to the cytoplasm for translation. In contrast, prokaryotic cells perform all three processes within the cytoplasm, often simultaneously, due to the absence of a nuclear envelope. Both prokaryotic and eukaryotic cells contain chromosomes, but they differ structurally. Prokaryotic chromosomes are typically single and circular, though some species may possess two or three chromosomes. Eukaryotic chromosomes are linear and exist in multiple copies. Both types of cells contain transposable elements, sequences of DNA capable of moving within or between DNA molecules, influence genetic diversity and genome evolution. Plasmids, found primarily in prokaryotes, are small circular or linear double-stranded DNA molecules that replicate independently of chromosomal DNA. While not essential for basic survival, plasmids can confer advantageous traits such as antibiotic resistance or virulence, enabling pathogens to attach to host, colonize tissues, and produce toxins. Some plasmids encode bacteriocins, proteins that inhibit or kill closely related bacterial strains, providing a competitive edge in microbial communities. The semi conservative nature of DNA replication ensures that each daughter DNA molecule contains one original parental strand and one newly synthesized strand. This process begins at a specific origin of replication where origin binding proteins open the DNA helix. Helicase unwinds the double stranded DNA, forming a replication fork, while single stranded binding proteins stabilize the unwound strands to prevent reannealing. A loader protein assists in the placement of two helicases, one on each strand, oriented in opposite directions. Primase synthesizes short RNA primers on the template strands, which serve as a starting point for DNA polymerase III, the primary enzyme responsible for synthesizing new DNA. The leading strand is synthesized continually in the 5' to 3' direction because a 3' hydroxyl group is always available for the addition of nucleotides. On the lagging strand, however, replication is discontinuous. Multiple primers are laid down and short fragments of DNA, known as Okazaki fragments, are synthesized. Now, these fragments are eventually joined together to form a continuous strand. As replication proceeds, DNA polymerase I replaces RNA primers with DNA using its 5' to 3' exonuclease activity. DNA ligase then seals the remaining nicks between DNA fragments by forming the final phosphodiester bonds. These enzymatic coordinations ensure the accuracy and completeness of DNA replication, maintaining genomic integrity across the generations. Together, these molecular processes illustrate the complexity and precision of genetic information flow and protein synthesis. Understanding these mechanisms provides fundamental knowledge for exploring cellular function, microbial behavior, and the molecular basis of life in microbiology. DNA replication is a highly coordinated and essential process in all living organisms, ensuring the accurate transmission of genetic information from one generation to the next. This process begins with the double stranded DNA molecule, which is composed of two complementary strands arranged in a double helix. During replication, DNA follows a semi conservative model in which each of the two resulting double helixes contain one parental strand and one newly synthesized daughter strand. The original strand, also called the template strand, guides the synthesis of the complementary strand, preserving the genetic code. At the core of DNA replication is a suite of enzymes, each with specific roles. DNA polymerase III is the primary enzyme responsible for replicating chromosomal DNA in prokaryotic cells. It adds nucleotides to the growing DNA chain by reading the template strand. DNA polymerase I plays a less prominent role in the elongation but is crucial in removing RNA primers and replacing them with DNA. DNA polymerase II, 4, and 5 are mainly involved in DNA repair, correcting damages that might otherwise compromise genomic stability. For replication to begin, a short RNA primer must first be synthesized. This primer, laid down by the enzyme primase, provides a free 3' hydroxyl group for DNA polymerase III to begin nucleotide addition. These RNA primers are later removed and replaced with DNA to form a continuous strand. Initiation of DNA synthesis begins at a specific location on the DNA called the origin of replication. Origin binding proteins recognize this site and open the double helix. Loader proteins then assist in binding two helicases enzymes, one on each strand, oriented in opposite directions. Helicase unwinds the DNA at the replication fork, creating a single stranded region that are immediately bound by single stranded binding proteins to prevent the strands from reannealing. Just behind the helicase, two primases and two DNA polymerases bind to the exposed strands to initiate replication. The replication of DNA proceeds in two distinct manners on each strand due to their antiparallel orientation. The leading strand is synthesized continually in the 5' to 3' direction, with DNA polymerase 3 following the helicase and adding nucleotides as the DNA unwinds. This process only requires one RNA primer. In contrast, the lagging strand cannot be synthesized continually because its orientation does not allow for a straightforward 3' hydroxyl to initiate synthesis in the same direction as helicase movement. Instead, primase lays down multiple RNA primers at intervals, allowing DNA polymerase 3 to synthesize short DNA fragments known as Okazaki fragments. These fragments are eventually joined into a continuous strand. The completion of DNA synthesis involves the removal of RNA primase and the joining of DNA fragments. DNA polymerase 1 removes the RNA primers using its 5' to 3' exonuclease activity and replaces them with DNA. DNA ligase, which plays a crucial role in DNA repair, seals the remaining nicks between the adjacent DNA fragments by forming the final phosphodiester bonds. DNA replication is bidirectional, meaning that it proceeds outward in both directions from the origin of replication. A large protein complex known as the replisome orchestrates this process, pulling the DNA template through it while synthesizing new strands. Within the replisome, helicase and primase form a subcomplex called the primosome. As replication continues, two replication forks move forward or move towards each other until they meet at the terminus region. Here, a protein called TES binds to a specific DNA sequence known as the TER site, effectively halting the replication forks. After replication concludes, the two newly formed DNA molecules remain connected and are separated during cell division by FITZ, a protein involved in cytokinesis. Accuracy in DNA replication is vital, and the cell employs proofreading mechanisms to minimize errors. DNA polymerase 3 is capable of inserting the correct nucleotide bases during synthesis, but if a mismatch occurs, proofreading mechanisms are activated. Both DNA polymerase 1 and 3 possess 5' to 3' exonuclease activity that allows them to excise incorrectly paired nucleotides. Once the mismatch is removed, DNA polymerase can attempt to insert the correct base again, maintaining the integrity of the genetic code. This intricate and highly regulated process ensures that cells can replicate their genomes accurately and efficiently, forming the foundation of cellular function, inheritance, and overall genomic stability in all forms of life. Fill in the blank worksheet. Here's the instructions. Fill in each blank with the correct term based on your understanding of molecular biology concepts. Number 1. The sum total of all genetic elements in an organism is called its blank. 2. DNA strands are held together by blank bonds between nitrogenous bases. 3. A blank consists of a nucleoside and a phosphate group. 4. Blank are enzymes that insert or remove supercoils from DNA. 5. In transcription, the enzyme blank polymerase synthesizes RNA from a DNA template. 6. In prokaryotic cells, all 3 stages of genetic information flow occur in the blank. 7. Blank are small DNA molecules that replicate separately from the bacterial chromosome. 8. During DNA replication, blank synthesizes short RNA primers to initiate DNA synthesis. 9. The blank strand is synthesized continually in the 5' to 3' direction. 10. DNA blank seals the final phosphodiester bond after RNA primer is removed. Here's multiple choice worksheet. Here are the instructions. Circle the letter corresponding to the best answer. Number 1. Which of the following is composed of a nucleoside and a phosphate group? 2. DNA strands are arranged in opposite directions. This arrangement is called. 3. What enzyme introduces negative supercoil into the DNA? 4. Which RNA type carries amino acids to the ribosome? 5. What occurs during replication? 6. In eukaryotes, where do replication and transcription occur? 7. Plasmids typically carry genes that. 8. Which enzyme adds DNA nucleotides to the RNA primase during replication? 9. What happens at the origin of replication? And 10. The lagging strand forms in small fragments because. Well, thanks for joining me in today's session. If you haven't grabbed your ebook yet, then what are you waiting for? Go to the description box and click that link. I'll see you in the next interactive tutoring session. Let's microbiology it.